So recall that the plasma membrane was described as a selectively permeable barrier. This means that some substances can easily cross the membrane, while others are prohibited from crossing. So now let's see how substances are able to cross the membrane. There are two general mechanisms. There is passive transport and there's active transport. Passive implies that substances can cross the membrane without any energy input from the cell. Meanwhile, active transport requires that the cell invests cellular energy, usually in the form of ATP, in order to move a substance across the membrane. So let's briefly compare the two, and then we can unpack some of these terms a little bit later. So passive transport requires no energy input from the cell. It is driven entirely by an energy called kinetic energy. The cell does not impart kinetic energy in any way to the substances that are moving. Instead, kinetic energy is simply the energy that the moving molecules have at the molecular level. In passive transport, substances move from areas of high concentration to areas of low concentration. And this is referred to as moving down or with a concentration gradient. We'll look at this a little bit more in just a minute. A good analogy for this is water moving down a hill with the force of gravity to a lower elevation. So on the other hand, active transport is going to require energy input from the cell, usually, as I mentioned previously, in the form of ATP energy. And with this energy input, the cell has the ability to move those substances against concentration gradients, meaning that a substance will move from an area where that substance is low in concentration to an area where that substance is high in concentration. Another good analogy for this is water moving up a hill, which it can only do so if energy is used to move that water, like the energy that is used to power a pump that will pump the water up the hill against the force of gravity. So let's focus on passive transport for a moment. There are two passive transport mechanisms. We have filtration and diffusion. So filtration is the process by which a substance is pushed across a membrane by a pressure or a force. A good example of this is production of tissue fluid, which is also known as interstitial fluid from the blood. So the pressure in our blood pushes fluid out of the thin walls of the body's capillaries, and it pushes that fluid into the space between the cells of that particular tissue. The other transport mechanism is called diffusion. And diffusion is the random motion of molecules from an area of high concentration of that particular molecule to an area of low concentration. So in this diagram, the substances that are dissolved in our fluid are referred to as solutes. So things like sodium, potassium, calcium, and glucose are all good examples of different types of solutes. So as I mentioned earlier, diffusion is not driven by energy from the cell, but it is driven by another type of energy called kinetic energy, which is the energy of motion at the molecular level. So now that we have described the basics of diffusion, we can examine a few different diffusion mechanisms. There are two major mechanisms, and both of these depend on whether the molecule that is diffusing is hydrophobic or hydrophilic. That is, it depends on the chemistry of the molecule that's moving. So recall that hydrophobic molecules do not dissolve easily in water, while hydrophilic ones do. Molecules that are hydrophobic, such as lipids, can move by simple diffusion, while substances that can dissolve in water will move by a type of diffusion that's called facilitated diffusion. So simple diffusion occurs with substances that are hydrophobic, or even some gas compounds, such as in this example we have oxygen and carbon dioxide, which also have the ability to move by simple diffusion. So as a whole, simple diffusion is a type of diffusion where molecules can diffuse directly across the plasma membrane. And keep in mind that this is still diffusion, so substances are moving from areas of high concentration to areas of low concentration. So in a real-life example, oxygen tends to diffuse from the extracellular fluid, such as the blood, into the cytosol of the cells. Cells are almost constantly using oxygen to make ATP energy, so oxygen concentrations in the cytosol tend to be pretty low, whereas in the plasma of the blood, which is an extracellular fluid, they tend to be a little bit higher. The opposite is true for carbon dioxide, or CO2, which is a gas that's produced when glucose is metabolized in order to make ATP energy. So we see that CO2 concentrations are generally very high in the cytosol, and they diffuse out of the cells into the extracellular fluid, where they're usually picked up by things like the bloodstream and carried to the lungs. A few examples of some hydrophobic compounds include steroid-based substances, because steroids are based off of the cholesterol molecule, and cholesterol, as we know, is a lipid. Certain vitamins, like vitamin A and vitamin D, are lipid-soluble, as well as some hormones are lipid-soluble, and they do have the ability to cross the plasma membrane by simple diffusion. These hormones include thyroid hormones as well as steroid hormones. And because steroids and steroid hormones are cholesterol-derived, they are able to cross the plasma membrane by simple diffusion. Steroid hormones include things like estrogen and testosterone, which are sex hormones, as well as some of the hormones that are manufactured by the adrenal gland. So now that we have seen simple diffusion, let's explore a little bit of facilitated diffusion. And so within facilitated diffusion, we have two types. The first is going to be a type that's called channel-mediated facilitated diffusion. And channel-mediated facilitated diffusion makes use of protein channels in the membrane. The membrane protein contains a water-filled channel at its center, and it allows for the passage of very small hydrophilic compounds, such as sodium and potassium, or even water itself can move by channel-mediated facilitated diffusion. The channels themselves are very selective about what can go through them due to the size or the diameter of the opening in that channel. So only very small elements can get through the smallest diameter openings, while slightly larger elements or molecules require larger channels to pass through. Keep in mind that this process is still diffusion, so substances, once again, are moving from regions where they are high in concentration to where they are low in concentration. Water also moves across the plasma membrane by facilitated diffusion. In this case, the process is typically referred to as osmosis. Protein channels that are called aquaporins permit the diffusion of water either into or out of the cell. Another type of facilitated diffusion is called carrier-mediated facilitated diffusion. And this type of transport is used for relatively large hydrophilic substances, such as amino acids or sugars. Even some carbohydrates can move by carrier-mediated facilitated diffusion, so things like that. Protein carriers that are involved have a very specific shape for the specific substances that they transport across the membrane. And the carrier works by binding the substance and changing shape in order to release that substance on the other side of the membrane. So in this example that I have for you here, we have a glucose carrier, and this glucose carrier has a very specific shape for a glucose molecule, and when that glucose binds, the shape of that carrier changes to move glucose across the middle, the core of that channel, and into the cell. So in this example, kind of backing up again, glucose is typically moving from the blood plasma, where it's in high concentration, into the interior of the cell, where it's typically in much lower concentrations. And the reason for this is that glucose is typically at low concentrations inside the cell because the cells are very rapidly using glucose, metabolizing it, harvesting the energy from the breakdown of that glucose to make ATP. So we've just seen that some substances and materials can move across the membrane without any energy input by the cell. And these are the passive transport mechanisms, like the two different types of diffusion, as well as the osmosis that we discussed, the movement of water. So the cell can also make use of active transport in certain situations when moving substances across the membrane. And so, however, unlike passive transport, active transport requires the cell to invest energy in that type of transport, typically in the form of ATP energy. So active transport typically makes use of protein pumps that are powered by ATP energy. The protein pumps of active transport will typically move substances from areas where that substance is low in concentration to areas of high concentration. And this is the opposite of diffusion, which is why active transport requires an input of chemical energy, like ATP, from the cell. So before ATP binds to a pump, the pump is in an inactive state, and it does not move substances across the membrane when it's in that inactive form or configuration. Once ATP is bound to that pump, ATP is then split, releasing energy, and that energy that's released is used to power the pump. And so from this release of energy, the pump is activated. It's going to move that specific substance that it's designated or designed to move across the membrane. Meanwhile, we see that ATP has lost one of its phosphate groups. And in doing so, it becomes a molecule called ADP, which stands for adenosine diphosphate. The phosphate group that was broken off of it will remain on the pump for a short time, after which it's ejected from the pump, and a new ATP molecule will be able to bind to that. So one example of an active transport mechanism or an active transport pump is called the sodium-potassium pump. As its name implies, the function of the sodium-potassium pump is to move sodium and potassium across the plasma membrane. However, it's going to move these two substances in opposite directions. The pump will move sodium out of the cell while moving potassium into the cell. First, we see that 3 sodium molecules will bind to specific sites on the pump, as well as binding 1 ATP molecule at an ATP binding site. This ATP that binds to it will soon be split. The energy that will be released from that splitting of the ATP will be used to change the shape of the pump. And in doing so, it's going to move those 3 sodium out of the cell. And shortly after that, 2 potassium molecules will bind to their own sites on the pump. The pump will revert back to its original configuration and release those 2 potassium into the cell. So we see that the pump in item 4 here on this diagram is now back to its original configuration, and it's ready to bind a new ATP and new sodium. We'll see later on that these pumps are really critical for establishing an electrical charge across the membrane of most of our body cells. And this electrical gradient, or this electrical charge that we see, is really important for the electrical signals that are created by muscle cells as well as the neurons of the nervous system.